Okay, so I'm really excited because the Cyberteachers channel has asked me to go and do some food reviews, which is great because I love free food. So here's the thing, it turns out they wanted me to do some biological tests on food, not actually getting to eat it. Gutted, but let's take a look anyway. Okay, so there's going to be 4 things that you need to test in this required practical. There's going to be carbohydrates, which are split into 2 groups, which are simple sugars, like glucose, and starch, which are complex sugars, lots of glucose molecules joined together. We're going to look at lipids as well, which are fats and oils, and we're going to look at protein. Okay, so for all your food tests, you're going to need to know a couple of things. First, you're going to need to know which chemical to use. Second, you're going to need to know what colour it is before the test. Thirdly, you're going to need to know what colour it is after the test. Okay, so let's test for simple sugars. We've got Benedict solution is blue before, nice bright blue. If it's blue afterwards, then there weren't any sugars in it. If it changes colour to a brick red colour, then it will show that there's a lot of sugars in it. In actual fact, this can change into yellow and green to show different concentrations of sugar, but for GCSEs, we just need to say that it goes from blue to brick red to show that it's got simple sugars. Let's do a quick test. Okay, as a colour standard, I've got some pure glucose here. If there's not any sugar in here, then there's not going to be sugar anywhere. So what I'll do is I'll take some of my glucose solution, add it to a test tube, and then add a few drops of Benedict solution. Okay, and I've added enough to show you that it's blue to start with. And the next thing we need to do with Benedict solution is to put it in a water bath. So I've boiled a kettle and we're going to use that. You could use a normal water bath, or you could use some a Bunsen burner with a flame. And as you can see, this glucose solution has changed completely from blue to a nice bright reddy orange kind of colour. Now let's prepare some food samples. So here for this test I've got some sweets that should contain glucose, and I've got some cheese here which might contain some but not as much. When preparing a food sample, you need to take your sample of food and place it into your test tube. Once placed in your test tube, you need to add some water to it. And then you need to give it a bit of a crush to try and release some of the insides. And these sweets are quite hard, so it's hard to crush them, but I'll do my best. With colour changes on things like sweets, it's hard to see the colour change sometimes, so ideally what you'd want to do is to drain the water off, and that's what the exam board will be looking for, and just test the fluid. And so as you can see, I've added some Benedict solution and I'm going to place that in our water bath. And as you can see, I've prepared some cheese as well, so that'll go in our water bath as well. Okay, so I've taken our samples out of the water bath, and here we have them. We've got the glucose, it's bright orange, pure glucose. We've also got here the sweets, clearly not as much pure glucose in it, but it's clearly not blue anymore. And then we've got the cheese, which has stayed blue. It had 5 minutes in the water bath, and that's all you need to give it. So the next thing we need to test for is starch, complex carbohydrate, lots of glucose joined together, and we use iodine for this. Iodine is a browny orange kind of colour depending on the concentration, and some people might say yellow, but this is quite a subjective thing. So let's have a look at what happens if we add iodine to a pure starch solution. Okay, and we can see that it went black blue. in colour when we added it to it. Now, in order to do food tests, we could use the boiling chips again, or we can use a spotting tile or a dimple tile like this in order to put our food samples into. So I'll show you a close-up of some of the food samples that we're going to do. Okay, so I've got 3 different types of food here that you can see. We've got some biscuit in here, which should contain carbohydrates. We've got some potato here, which should also, and we've got some cheese here. And so I'm just going to add some water to each one of them. And now the water's been added, let's add some iodine to the biscuit, to the potato, and to the cheese. You can see that the biscuit's gone a blue-black colour, you can see that the potato's got blue-black on it, and you can see that the cheese has stayed orangey. It shows that there is starch in the potato and the biscuit, but not in the cheese. Okay, the next thing we need to test for is protein. For this we use Biuret's solution, which is a mixture of sodium hydroxide, which is a colourless solution, and copper sulfate, which is a light blue solution. When you mix them together, it will make it look blue. If it goes purple, then it's a positive test for protein. We're not going to do some food tests on this because I've shown you how to prepare samples, crush them, add water, etc. So I'm just going to add this to pure gelatin, which is definitely a solution containing protein. So let's see what happens. And so adding the blue copper sulfate and the colourless sodium hydroxide, we can see that there's been a purple colour change, purple lilac, the exam board will be looking for. This is positive for protein. Okay, last thing we're going to test for is lipids, that's fats and oils. And in order to be positive, to show you the colour change on this, we're just going to use some oil that I've got prepared here. And there are 2 ways of doing it. The first one's with Sudan III solution, which is this red solution. Okay, and what you need to do is you need to add just a couple of drops of Sudan III to your food source, and if a red band forms at the top, then we can see that it's positive for lipids. Let's see. So you can see a red band at the top because oils and fats are immiscible. Okay, the other way to test for lipids is using ethanol, which is a colourless solution, and if we just take it and add it to our food sample, which in this case again is oil, we'll just add it's meant to be about 2 centimetres cubed that you add to it, and you can see that it is, well, that will do, but we can see that it's split and immiscible, and then we need to add some water to it, and if it goes cloudy in appearance, then there was lipid present. Give it a little shake, and it's gone cloudy. It's no longer clear, it is still colourless, but it's gone cloudy, which shows that there was lipid present. Okay, and here we have our food tests. And so we started with simple sugars, simple carbohydrates, which went brick red when we added Benedict's solution to it, which was blue to start with. Then we looked at complex carbohydrates, starches, which went a bluey black when we added iodine to it, which was kind of like a yellowy brown to start with. Then we looked for protein and we added Biuret's. It goes purple if it's, or a light look colour, for a positive test, and we added Biuret's, which is sodium hydroxide and copper sulfate solution, blue and clear. And then we had a look at lipids, and we've got a red band at the top with lipids when we add Sudan III solution. And then finally another test for lipids, we added ethanol, and ethanol is clear, but we've got a cloudy kind of emulsion at the bottom there to show the presence of lipids. Why not check out some more Cyberteachers videos, or maybe our short 60-second versions, our Cyber 60s?
If you liked this CyberTeachers video and found it helpful, please don't forget to like and subscribe, and we'll keep the CyberTeacher content coming.